Hello everyone, Jono here. So I just shared this short showing what happens when you drop some duckweed that has been soaked in isopropyl alcohol into water. You get this crazy running around effect. So I discovered this a while back, and I thought in this longer video I would talk a little bit about what's going on there, but also the backstory for why I even had this lying around and how it was found. So if you're interested in just the short, this is a brand new channel. I don't know how either shorts or longs work, but stick around if you'd like to see some of the backstory. So this all started when a really cool guy who does a lot of DIY biology and science, Sebastian, shared there's model organisms everywhere for those to see. He found some duckweed in a ditch by his house. And I was like, wait, I've got, ponds by my house. I've seen this duckweed. And so I grabbed some, and it came along with lots of other bits of life. But the cool thing about duckweed and the reason it makes a good model organism is that it replicates really fast, right? You get these leaves and they sort of bud, add extra buds, and split out. So I actually took a time lapse. You can see when you speed it up, the share of volume increases quite rapidly. And if you watch closely, you'll see individual little ones cleaving in two and then having these little clonal buds that spring out from them, although it does also sometimes reproduce sexually. Anyway, so it's pretty easy to grow, and so I collected some and I started growing it. You can see here's the first few little fronds that I put in, and not that long later, maybe a week or two later, you end up with a much larger volume. So the reason I was interested in it is I thought, well, this stuff's cool, and also I've been looking for like a good canonical example of the kinds of AI-assisted science that I'm excited about. There's been lots of talk of AI for science, and for me, the part about that that's interesting is the human creativity combined with the like encyclopedic knowledge of these AIs. So the way I set out to frame this as a fun little experiment and blog about it was, can we use LLMs as kind of general optimizers to help me figure out an ideal mixture of nutrients for these plants? And so I set up these different tubs, and in each one there's going to be some number of duckweed plants, right? And then we're going to count how many fronds there are. We're going to repeat that over time. And sure enough, you can, see some variation, but I didn't even know where to start. So I just went to the Encyclopedia of All Human Knowledge, loosely remembered and often hallucinated, ChatGPT, and fed in, this is the target, this is how I'm growing it, it's sitting by a light this distance apart, etc., suggest some nutrient mixes. And so then it was able to recommend, I had some different stock solutions on hand. So MaxiGrow, plant hydroponic nutrient, just general mix, and then also some potassium dihydrogen orthophosphate, KTP, a couple of other bits and bobs to tweak the pH and so on. And so this was the process. We would put out the same number of fronds in each little trough. The LLM would suggest the different mixes, just water as a control, 6 milliliters of S1, suggest some various mixes, and then over time I could track that. I could see how the number of fronds increased given the different mixes and pick the one that was optimal. So that was why I had all the duckweed lying around. But in the same time as those tubs were going on, the main lot was also growing. So I just ended up with this tub full of duckweed. And then rather than throwing it out straight away, I thought, well, the other thing that I'm a little bit obsessed with at the moment is fluorescence. So if I mush some of this up in isopropyl alcohol, I can extract, you can see the chlorophyll coming into the solution there. Then when you hit this with the UV light, it fluoresces bright red. Even the plants themselves also fluoresce red under deep ultraviolet light, or in this case, a very blue to violet laser. So I wanted to extract out the chlorophyll to get some pretty glowy colors. No real scientific objective other than, hey, it looks looks pretty cool. But as I was then doing the washing up, I noticed the pieces that I was throwing away in the sink starting to whiz around, and that was why I took this first video and noticed it. So the motion here is it looks like surprisingly organic and lifelike and fun, right? Like a lot of cells running around. But I'm pretty sure at least just driven by the changes in surface tension. I guess some people call it the Marangoni effect. This duckweed has absorbed some of this isopropyl alcohol. It's slowly leaking out. It's the same thing as like a soap boat, if you've ever seen that experiment. And so to double check that we understand that and to see if we can simulate it, because that's one way to understand it, I thought I would attempt to then close off this video with a quick little AI-assisted computer simulation of what I think is going on and see if we can match the motion. And so the framing that I'm going to set up, let me just switch to pen, is that we're going to represent our water surface as a grid, right? And in each of these points, we're going to keep track of the surface tension. And so by default, we'll set it all to 1. And then for every duckweed frond, for every particle, we're going to represent the way that it's leaking alcohol as being slightly different, right? I mashed these up. They're going to have different like cuts and tears. And so the way we're going to represent that is with kind of like a little 3x3 grid or kernel to show kind of like which directions is the alcohol leaking out most preferentially, right? And so if we said there's none leaking out this side, but there's a little bit leaking out here, and a little bit more leaking out here, right? You can imagine that the surface tension is going to change more on this side where this extra stuff is leaking out. And so for every little particle on our grid, what we're going to do is we're going to take the base surface tension, we're going to subtract the little kernel that we've written, and then we're going to calculate as a result, like which direction is this going to move. And also, if there is more push on one side than the other, which direction is it going to rotate, right? And so this is the setup that I've described. We want to have this constantly changing grid. And then once we've changed the surface tension, right? So now we've applied our little update. This is minus some little delta, right? Over time, this is not just going to stay changed. The liquid is a liquid, right? There's lots of motion and diffusion going on. And so we're going to then also do a diffusion step where we're going to update the surface tension at every point based on the neighbors. So there's going to be a slow spreading out of any changes. So if you made the surface tension really low in one spot, the next step it would be kind of low in that region. The next step it would be faintly lower in the rest, and it'd spread out that way. So I've asked two rival AIs to set up the simulation based on that description. You can see here's my dictated description of exactly what I want. Here's the code. And if we run this, we can now click to add our little duckweed particles, and you can see somewhat reminiscent motion compared to the physical one that we did. And you'll notice like sometimes they definitely seem to interact, right? They steer together and they bump off each other. There's no collisions happening here. The only means of communication is via the changes in surface tension that they both cause and the motion that engenders in each one as a result. So it's really fun to play around with. I also have an artifact that I can share, just a different version of the same thing by Claude 4.5 Sonnet instead of ChatGPT. And you can tweak how much the rotation is affected. You can tweak how many particles you add and so on. But yeah, it's fun to see like this very interesting, organic looking, emergent behavior, right? Like it's very hard not to attribute agency to these individual little dots moving around. But in actual fact, this is just driven by a relatively simple set of rules of how each one affects the neighborhood around it and how it moves in accordance with that neighborhood around it. Just surface tension and slow leakage of one liquid into another. Anyway, so that's my little duckweed odyssey. I hope you found that interesting. Do check out the channel. I'm going to try and do a lot more of these short form videos, just sharing fun things that I've observed, interesting pieces of nature, and you'll notice this theme of simple rules leading to complex emergent patterns cropping up again and again and again. Thank you so much for watching.